Okay, so good afternoon, ladies and gentlemen. Thanks for tuning in today. My name's Francis. I'm a PhD researcher at the Institute for Risk Uncertainty at the University of Liverpool, with a keen interest in emerging industries. So I'll be your host for today. So this is the first session of our conference series on emerging industries. We'll be exploring these new industries that are beginning to design, build, and test novel solutions to our modern-day world challenges. They present an array of opportunities, but may also create threats and risks to society, individuals, and environment that we should explore carefully. Today, we'll be looking at nanotechnologies and their associated opportunities and risk trade-offs. The present and future applications of nanotechnologies reveal a universe of possibilities: invisible particles that fight cancer cells, faster microprocessors that consume less energy, batteries that last much longer, just to name a few. I even remember myself writing about nanotechnologies for aircraft in my personal statement, and that got me into university. On behalf of the University of Liverpool's Institute for Risk Uncertainty, I'm really delighted to welcome two accomplished researchers, Professor Vicki Stone from Heriot-Watt University and project coordinator of the Gracious Project, a framework for streamlining risk assessment for nanomaterials, and Dr. Igor Linkov from the US Army Engineer Research Development Center, and he's a representative at the National Nanotechnology Initiative, NNI, for the US Army. I'll be introducing them both with a bit more detail shortly. The agenda for today is a framework to streamline nanomaterial innovation and risk assessment, talking about the Gracious Project. So that'll be from Professor Vicki Stone, starting in 2 minutes. And following that, we'll have Professor Igor Linkov on a personal reflection on 20 years of nano risk assessment and relevance to emerging risks. And that'll be starting at 3:05. Then to wrap up, we'll have a 30-minute open discussion on how useful are alternative models and methods in assessing the risks of nanomaterials, which I'm really looking forward to. So without further ado, let me introduce a little bit about Professor Vicki Stone. She's the director of the Institute of Biological Chemistry and Biophysics and Bioengineering at Heriot-Watt University. She was the holder of the Royal Society of Chemistry Toxicology Award in 2015-2016, and is the editor-in-chief of the Journal of Nanotoxicology for 6 years between 2006 to 2011. She also published over 180 publications pertaining to particle toxicology over the last 25 years and is recognized by Clarivate Analytics as one of the top 1% of all researchers in the world, with the most cited publications in the field of pharmacology and toxicology. And she was also a partner in 3 EU-funded projects. So as I mentioned, the EU Gracious Project, Petrols, and BioRima. So thank you very much and enjoy Vicki's talk.
Thank you. You want me to go ahead and share my screen?
Please, can you?
Yeah. Okay, thank you very much for that introduction and for the invitation to speak at this meeting today. I realise we have quite a varied audience, so I'll try and make my talk as accessible as possible. So today I'm going to focus on one of our current projects called Gracious, for which I'm the coordinator. So this is a European project, and it's making a framework that's helping people to streamline nanomaterial innovation and risk assessment. So these are some of the things I'm going to talk about today. So I'm going to give you an introduction to one of the methods that can be used for streamlining risk assessment, and that's grouping and read across. And then I'm going to describe to you the Gracious framework and how it supports the grouping and read across of nanoforms. And I'll explain what a nanoform is in a minute. This will involve us going through a number of grouping hypotheses and then methods for generating evidence to test these hypotheses. And we will look at integrated approaches to testing and assessment, or IATAs, to do that. We'll talk about how we can generate thresholds to decide whether something is in a group or whether it's outside of a group. But we'll also look at a slightly easier way of grouping, and that's to assess similarity and we've got some both qualitative and quantitative methods for that. We'll also talk about the need to ensure that you have good quality data and complete data sets for conducting such activities. And then I'll finally finish off by telling you how you might access the Gracious framework if you're interested. So the Gracious project started in January 2018. It finishes later this year, and we have 23 partners who are scattered across Europe and 2 of them in the US. And so the aim of the project, as I've said, is to make this framework to support risk considerations for nanomaterials. And it does this by allowing people to apply grouping nanomaterials together in a logical way so that you can read across or copy data from data-rich nanomaterials to those that lack lack information that you need for risk assessment. I'm going to use this term nanoforms through my talk. So first of all, let me explain what a nanomaterial is. So a nanomaterial is an object or a particle which has 3 dimensions in the nanoscale. So that means below 100 nanometers. And also some of these materials will have 2 dimensions in the nanoscale. So they might be shaped like a rod or a fiber, for example. And then these nanomaterials are available in a wide range of different physical and chemical properties. They could be metals, metal oxides, polymers, carbon-based, almost any composition that you can think of. And they come in a variety of different shapes with coatings, without coatings. So they're really a diverse array of substances. And people use them because when you create materials at the nanoscale, they tend to change their properties that you can exploit in commercial products. So for example, gold at the nanoscale is reactive, it's a catalyst, and also you can use it for imaging. It's used in medicine for targeting tumors and killing tumor cells. And it's used for imaging tumors in the body. So something that would normally consider as a gold inert material, when you put it in the nanoscale, has these new properties that you can exploit. So there are lots of different nanomaterials available made from different substances. And recently, the European Chemicals Agency, or ECHA as they're known, this is the group in Europe that regulates the sale and use of chemicals within Europe. They've introduced a new term called nanoforms. So nanoforms are different shapes, different sizes of the same substance. So you might have titanium dioxide as a nanomaterial, and then there might be 10 different sizes, 3 different shapes, and they would be different nanoforms of that nanomaterial. So I'm frequently going to use the term nanoforms today because grouping is often used for the same chemical substance, but not explicitly for the same chemical substance. So that's why I'm going to use that term today. Okay, slides are moving slowly. So when you the conventional approach to a risk assessment is to consider each substance on a case-by-case basis, but this is really expensive, it's time-consuming, and from a toxicology perspective, it uses a huge number of animals, which is not ethical and it's not sustainable. So what you can do is you can take your different nanomaterials or nanoforms or different substances, and you can group them together in a logical way. And most people think about grouping in terms of their physical and chemical properties. So what are they made from? What do they look like? But you can also group them based on their toxicology or their ecotoxicology. And in fact, the REACH regulation, which is the regulation in Europe that controls chemical usage and marketing, states that you can, if you have substances whose physicochemical, toxicology and ecotoxicology properties are likely to be similar or follow a regular pattern as a result of structural similarity, they can be grouped. So basically, if they're made from a similar substance, they have a similar shape, similar properties, and similar hazards, then you can group them. So Gracious is a project that has generated a framework that allows you to logically group your nanomaterials to get together. It gives you more guidance on how to do this, more detail in an evidence-based way. And it provides not only qualitative expert judgment for assessing similarity, but it also provides a number of quantitative tools for assessing similarity as well, which is useful when you're putting together something like a document for a regulator. So in order to construct the Gracious framework, we consulted the literature. So we looked at all of the projects that had been conducted in a similar area before, as well as the regulation, combined these together into a draft document. And this allowed us to combine regulatory and industry and academic opinions in this area into a framework that would meet all of the requirements of those three different communities. And then we shared this with a wide range of stakeholders to get their feedback and to improve the clarity of the framework, which I'm going to show you today. So here's the grouping and read across framework that's been generated by Gracious. It's been published last year in the journal Nano Today. And I've got two diagrams which I'm going to show you. This is the simple form of the Gracious framework, and then the next diagram is the same framework but with more detail included. So you would enter the framework using either a single nanoform that you're lacking data on. So you've got a nanoform, you want to use it in a particular application, or you want to design one to use in a particular application, but you don't have all of the data, all of the hazard data especially, that you need to do a risk assessment for that nanoform. Or it may be that you have a provisional group of nanoforms, so more than one substance there. Some of them may have the data you need for risk assessment, and some of them will be lacking that data. So you enter the framework into the basic information step. So in previous risk assessment softwares that other projects have generated, one of the criticisms have been that they ask for too much information. It's too complicated, the people give up and they walk away. So we have a basic information step, which just gathers the key information needed to get that person started. And this information allows them to generate a hypothesis to explain why their nanoforms can be grouped together. And this hypothesis takes into consideration the hazards and the phys-chem characteristics and potential exposure routes for humans or environmental compartments. It then provides some testing strategies that allow you to gather the data that you need to support or reject your hypothesis. And these testing strategies are tiered, so you can use simple tests in tier ones. These might be computer modeling in silico methods, or they could be simple cell models with just one cell type studied in vitro. Tier 2 would be more complex models, and then tier 3 would be animal models. And you use these sources of information to test the hypothesis. And if you can accept your hypothesis, then you can group your nanoforms together. And if you can't accept your hypothesis, you can either refine it, so it gives you enough information to understand how to refine your hypothesis, or it may be that you decide that grouping's not going to work for your particular nano material, and so you assess risk on a case-by-case basis. So the next diagram is the more complicated version of the Gracious framework. I'm not sure why it's so slow to move from one slide to the next. Okay, here we go. So it's exactly the same framework again, but now you can see we're starting again with the single nanoform or the provisional group, but now we have some information on the basic starting information. So we're asking for just 4 physical and chemical characteristics, so things like composition and impurities and size and shape. We're also asking for information that will help us to understand how that nanoform is likely to be released into the environment. So what are you using it for? How are you using it? When are you using it? So that we can work out what the likely release and exposure scenarios are. So that could be for humans in an occupational setting, could be for consumers, or it could be for an environmental consideration. And then also, why are you doing this grouping assessment? Is it because you want to generate a document for a regulator, or is it because you're in the early innovation stages of your new product and you want some information to make a quick decision about whether your the nanoform ideas that you have are likely to be safe or not? Once you have that information, then you can generate your grouping hypothesis. And the Gracious Consortium have generated 40 different predefined hypotheses that are available to the user, covering both human and environmental hazards. And we've used a template to generate these hypotheses, and this template is available to the user so that if the hypotheses we've generated are not suitable for your requirements, then you can make your own user-defined hypothesis. So once you have that hypothesis, you need to test it, and you test it using something called an IATA, an integrated approach to testing and an assessment. So this allows you to integrate different sources of information, existing information, modeling information, and testing information together. And once you've assessed your hypothesis, that means you've generated information and data which you feed into a data matrix. And this data matrix allows you to lay out all the evidence you have in one place in a logical way that allows you to assess whether your hypothesis can be accepted or rejected. And a data matrix is required by the European Chemical Agency for submissions to them for chemicals. Earlier on, I mentioned that you need to identify the purpose of grouping. This is important because that gives you an indication of the level of detail you need to generate at this stage and the level of certainty that you need in your data. So for a regulatory submission, you would need a higher level of certainty than you would for a safe by design application or an innovation application. So as I said, once you've gone through this process, you can decide either to accept your grouping hypothesis, to refine it, or to just go and do a risk assessment on a case-by-case basis. So once you've done your grouping exercise, you now have your group of nanomaterials or nanoforms, and within that group, some of those nanoforms should have data available, and these will become your source materials. They will provide the data that you need to fill data gaps for the materials in your group that are lacking data. So you already have a hypothesis for your group, so you can edit that hypothesis to make it suitable for read across. So your hypothesis might state why you can read across from your target to your source nanoform. Your data matrix contains the information that you have to support or substantiate that hypothesis so that you can then carry out read across. And ECHA has a number of methods that it puts forward for conducting read across. It could be that you just average the data and then use that average measurement. It could be that you take the worst case nanoform from your group and copy the data from your worst case to your target nanoform. It depends upon the data that you have and your personal choice. So I've mentioned a couple of times that the framework is not just for supporting regulation, but for supporting innovation. And I've mentioned this term safer by design. So this is a term that's being used widely in Europe at the moment. Then there are several projects on this, and this is trying to build safety into the early innovation stages of substances and nanoforms so that as you develop them, you take safety into consideration. This block of blue squares here is called the stage gate process, and it describes the process from early discovery through to the launch of a product, and the user would go through a series of gates at which they would make decisions as to whether to continue with the development of that product or not. And you can see that the basic information step of the Gracious framework could provide information which would support decision making at stages 1 to 4 of the stage gate process. And then the detailed step could take you as far as the final stages of launch, where you would need to consider submitting a dossier to the regulator. So we that's the overall structure of the framework. So now I'm going to focus on the grouping hypotheses. As I said, we have a template for these, and this template takes into consideration the purpose of grouping, which I've mentioned already, but also the context. So is this for a consumer? Is it an occupational setting? Is it for the environment? We also need to take into consideration the life cycle. So at what point in the life cycle of that nanomaterial or nanoform are we interested in assessing the risk? Is it during its manufacture? Is it during its use? Is it while it's sitting on the shelf in someone's house, or is it after disposal? And this is important because it feeds information to these three questions here: what they are, the physical and chemical characteristics of the nanoform that you need to consider, the where they go, so where they go in the environment, where they go in the body, and then what they do, so their hazards to humans and their hazards to environmental species. This template has allowed us to generate 17 human hazard hypotheses and 23 hypotheses for the environment. I'm just going to show you a couple of examples of these hypotheses now. The first one's the human one, the second one's the environment one. So they always start with the most relevant physical and chemical characteristics relevant to the hypothesis and the route of exposure, shown in orange, and then this blue text covers the hazard. So here we have a nanoform which is quickly dissolving. It's when it's inhaled, both the nanoform itself and any constituent ions or molecules that are released as it dissolves quickly in the body, they may contribute to the toxicity. So it means that the user needs to consider the nanoform and anything released from it when they're assessing their hazard. But there shouldn't be any concern for accumulation because it quickly dissolves. It's not going to accumulate in the body. But when you're assessing the toxicity, you need to think about where it's dissolving in the body and then what it's releasing in order to understand the hazard. For the environmental hypothesis, here we have one with an example of nanomaterials that are coated. Many nanomaterials have coatings on their surface in order to give them stability, to give them charge, to give them different physical and chemical characteristics that are useful for their application. But not all of these coatings are stable, they can be lost. So this is a hypothesis that looks at the stability of that coating, and it's suggesting that if the coating is lost when nanoforms are released into the environment and they're exposed to soil compartment, that they can be grouped if they lose it, and that they can be compared to nanoforms that don't have a coating. So for example, if you had a silica particle with and without a coating, you could group them together if that coating is lost in the soil compartment. So our hypotheses don't just focus on inhalation for human health, they also cover the gastrointestinal tract and the skin. And for the environment, they cover the aquatic, terrestrial, and sediment environments as well. So now we need to look at these integrated approaches to testing and assessment that have been developed to assess these hypotheses. So this is a generic IATA, as they're called. These IATAs have been proposed by the OECD. So the OECD is an organization that helps communities around the world, many international countries, to standardize the way that they do things, such as toxicity testing. And if you use these standardized methods from the OECD, it means you can compare data from one country to another. So we've used this OECD format for generating the IATAs, and these IATAs are a series of questions. They're a decision tree with yes and no options. These blue boxes are the questions, which we've called decision nodes. And behind each of these questions are a series of SOPs, standard operating procedures. These are standardized methods that you can use to assess that question. So each IATA is triggered by a specific hypothesis. So the IATA content will be tailored to the hypothesis, and it will include considerations of the likely route of exposure, the phys-chem characteristics of the particles, which target tissues or environmental compartments they are likely to come into contact with, and then the mechanism of toxicity. So this is important because it allows you to identify biomarkers of toxicity that are related to their risk. And if you say yes to all of the questions in the IATA, then you can group your nanoforms using this hypothesis. And if you say no at any point, then you would reject your hypothesis. We need some way of deciding if the answer is yes or no, and this can be achieved via thresholds, but thresholds are very difficult to justify, or by doing something called a similarity assessment, which I'll talk about later. And as I've said, these IATAs feed into a data matrix to support your decision making. So the IATAs don't just advise you on how to do testing, they allow you to take data from existing data repositories and the peer-reviewed literature. They allow modeling approaches to be included, and they allow testing. So that's one of the advantages of this IATA approach, is that they're versatile in terms of the types of data that they can accommodate. So you can then have a data matrix, which would have your different nanoforms and different endpoints. You could collect it together and identify whether your different materials can be grouped together or whether they cannot be grouped. So I don't want you to read this, but these are 10, I think, hypotheses that have been put forward for the oral route of exposure. So these are nanomaterials that might be ingested. And there are 4 types of hypotheses here, and these types of hypotheses are driven by how stable the nanomaterials are when they get into the gut. So the gut is a highly hostile environment with extreme differences in pH, lots of enzymes present, lots of chemicals present that can degrade nanomaterials. And so this is a key determinant of the hypothesis type. So we've got hypotheses that address instantaneous dissolution, quick dissolution, partial dissolution, and very slow dissolution. And so if we look just at the partial dissolution ones, we've got hypotheses that consider impacts on the gastrointestinal tract, hypotheses that consider translocation from the gut into the body, and we also have a hypothesis that considers impacts on the microbial communities that live in our gastrointestinal tract, which are essential for the health and function of our bodies. So you can start to see how you can create these different hypotheses and how important dissolution is in creating these hypotheses. And there are methods available to assess without using animals that can allow you to predict the dissolution and stability of nanomaterials in the gut. And so you would assess dissolution in saliva, in a stomach simulant, which would have a low pH, in an intestinal simulant, which would have a slightly higher pH. And there are different times. Sorry, my times have moved. This should be 5 minutes, 125 minutes, and then 155 minutes here. But you can see how you're simulating what happens in the body. And this protocol has been based on protocols that have been published by others and other projects. Here we've used this method to compare a number of different nanomaterials. We've got zinc oxide, silica, and titanium dioxide, and this is the half-life. So this is the time that it takes for half of your material to dissolve approximately in your experimental conditions. So the titanium dioxide is biopersistent. It doesn't dissolve. It has a very long half-life, whereas the zinc oxide dissolves relatively quickly in these conditions, and so it would not last for a long time in the body. So it would be one of these very quickly dissolving. Whereas the silica is intermediate in between, so it would be one of these partial or gradually dissolving materials. So you can use these kinds of experiments to help you to start to think about cutoff values, the thresholds that you would use in your IATAs. So this work and the work from the European Foods Safety Authority and various publications have been used to generate this IATA. So this is a full IATA for the gastrointestinal tract, and I'll just take you through it slowly. You don't need to read all of the detail. So we have 4 different hypotheses coming, or 4 types of hypotheses coming out of this this IATA. Here we have the instantly dissolving, the quickly dissolving, the gradually dissolving, and the very slowly dissolving. So at the top, before you get to the IATA, from the basic information stage, from the gracious framework, you would establish that the oral route of exposure is relevant. And so the first thing that you would need to do is assess dissolution half-life in gastrointestinal tract fluids, as shown in the previous slide. And so there are cutoffs here of 10 minutes, 10 to 2 hours, 2 hours to 60 hours, and more than 60 hours that allow you to put them into these instantaneous, quick, gradual, and very slowly dissolving groups. So once you've got those 4 groups identified, if they're in the instantaneously dissolving group, you don't really need to do any more testing. You can just go and find out about the toxicity of the dissolved substances that are released from these nanoforms and just read across to that substance, providing that that information is available. So there's no nano-specific hazards to consider here. Whereas the ones where the particles persist for a bit longer, even for the quickly dissolving and the gradually dissolving materials, you need to consider both the nanoform and the dissolving substance as shown in these diagrams here. So at this point, there's a potential for particles to be taken up into cells. So we need to consider dissolution inside cells, and we have a protocol for doing that. And again, we have cutoffs that are based on evidence in the literature of 48 hours to 1,440 hours or greater than 1,440 hours that allow you to identify whether your materials are not biopersistent, or highly biopersistent. So the longer they persist in the body, the greater their chances of causing hazard that are associated with the particle or nanoform structure. So once you have that information, you would then go on to assess the hazards using some of the mechanistic endpoints, which I'll show you in a minute. So it's quite a complicated structure, but the key way to break this down is to say, well, to start off with, we're going to focus on dissolution in the gut. Then we're going to focus on dissolution inside cells. And then once we know whether we've got nanoforms or ions present or a mixture of both, then we can assess the hazard. So that's how that IATA is structured. So that's for the oral route. The next slide shows you a different IATA, but this time for the inhalation route. I'm not going to go through it in detail, but it shows you exactly the same as in the previous slide. You need to consider the biopersistence of these materials or their potential to dissolve in lung lining fluid and in cells, and the hazards associated with that. If I can get the slides to move. I'm sorry, but my computer is hanging at the second. I just need to wait, sorry. I'm just going to escape from my presentation and go back in again to see if that helps. Oh, it wants me to restart the program. Oh no, let's wait for the program to respond. Okay, I think we're okay now.
At the moment, it's still on the same slide, but
yeah, it's
you can try and exit it if you like.
Yeah. It's asking me to restart the program. I'm really sorry.
Yeah, I mean, by all means, go ahead. I guess you have a lot of images in there. Maybe that's kind of what's slowing it down, possibly.
Okay.
On a humorous note, though, I noticed that Professor Scott has changed his nickname on the Zoom. Scott Surface something.
I think you might be multitasking.
Okay.
Possibly.
Sorry, I couldn't hear you. Could you say it again?
No, I was just wondering what's your new nickname. It says Scott Surface something.
That's Francis's fault. He picked that name. Sorry, I have competing noise here, so I'm trying to be mute.
Hopefully your computer is not crashing, Vicki, and
No, it's
you're able to
just
reopen
coming
it.
now.
Okay, perfect.
So I don't know what you can see at the moment.
We can see the slides, not full screen or presenter mode. Fingers crossed. Okay, we're on your view. Great.
Okay, but unfortunately we need to just, I think a quicker way is actually to scroll down the slides. Sorry. Okay, I'm skipping the slide where we had the problem, just, just in case it causes another problem. Sorry about that. Okay.
No
So as going back now to the oral IATA, and I mentioned at the bottom of the oral IATA there was a consideration of the hazards and that you needed to think about the mechanism of toxicity here. So this is an expansion of that part of the IATA, and it's to show you that it takes into consideration the physical and chemical characteristics of the nanomaterial, what impacts they're going to have on different structures inside the cell, whether they kill cells, whether they promote mechanisms that drive disease, such as enhanced immune responses, or whether it causes DNA damage that might lead to cancer, for example. So it asks all of these types of questions in there, and if the answer is yes, then you know you can support your hypothesis and you have an issue that you need to address, but it provides you with the evidence that you need to consider read across to substances as well that may have the information you need within your group. So this is a smaller version of the same diagram on the previous slide. So how do you make this decision? How do you decide whether your answer is yes or no? So behind each one of these decision nodes are a series of tiered testing strategies. So for each tier, there's an increasing level of complexity. Tier 1, simple. Tier 3, more complicated. For every tier, there's a requirement to review the existing data so that you make use of existing data wherever possible. For tier 1, if that data doesn't exist, then there are methods proposed in the tiered testing strategy that could be computer-based, chemical-based, or simple cell models in vitro. They could be more complex in vitro models. So for example, models of different tissues that have different cell types in place, or they could be animal models. And then for the environment, it could be tier 1 is a simple individual microorganism or an invertebrate or an algae, whereas tier 2 could be a repeated exposure over a longer duration, and tier 3 might be a mesocosm study where you have several organisms in the same conditions together. The level of information that you need depends upon the purpose of why you are grouping. And so the purpose, it doesn't mean that for regulatory purposes you would always go to in vivo models. What it means is for regulatory purposes you need more certainty to make a decision. You may get enough certainty from a tier 1 method, but it's likely that you might need to go to a higher tier method to get more evidence to increase your level of certainty. Whereas in innovation, you might be happy with just tier 1 methods for making your decisions. So here is a tiered testing strategy for the oral IATA. Along the top are the individual decision nodes, and below is tier 1, tier 2, tier 3. And you can see that each box is populated with some examples of methods that we would recommend. So we've used, for example, OECD methods from previous projects, methods in the peer-reviewed literature. Wherever possible, they're standardized methods to allow that mutual acceptance of data between different organizations. So this next slide or next series of slides I know is going to be of interest to the second speaker, Igor, because I'm now going to talk about thresholds and cutoffs and assessing similarity. And this is one of the big questions that people always asked us at the beginning of the Gracious project. Oh, stuck. What I might do is just come out of the presentation mode and present it so that you don't see it in full slide mode and see if that helps.
Vicki, you just announced the most interesting slide and it doesn't show up.
Yeah, it's a tricky one. I think the images are quite dense, so I guess it just can't hack it. We can see that, okay? Sorry, Vicky, you might be muted, possibly. Hello? For some reason, we can't hear you. Sorry, yeah, I think we can hear you now. Was it just me, or it might have been my headphones?
No,
I
I've just
couldn't hear as well.
unplugged my headphones and I'm just talking straight into the computer. So hopefully the audio is okay. I'm really sorry about these issues. Okay,
No problem. We can hear you perfectly fine though. Now.
Okay, good. Thank you. So, so fibers such as asbestos are a problem because they are thin enough that they have a small aerodynamic diameter when they're flying through the air so that they can be inhaled into the respiratory system. But they fly like a javelin, and so they have length, and this length makes them difficult to clear from the body. It makes them too big to be taken up by immune cells and cleared by immune cells, and it makes them too big to drain from little holes. This is the lung here. This is the pleural cavity around the outside, and there are holes in this pleural cavity that allow particles to drain into the lymphatic system and into your lymph nodes. And what happens is if you get this really small, narrow, but long fibers, they can be inhaled, but you can't clear them from the body because they're too big for the immune cells and they're too big to pass through these holes. And so they tend to accumulate in this cavity around the outside of the lung called the pleural space. They interact with the cells there, they cause damage to them, and that can lead to fibrosis, which is scar tissue formation, but it can also lead to cancer as well. And that cancer is known as mesothelioma. So on the basis of that knowledge, we generated an IATA that can address fibers. I'm not going to go through the IATA, but I just want to point out to you that there are 1, 2, 3 different decision nodes which all have thresholds. They have thresholds for dissolution, which we've talked about already, in the lung lining fluid and inside the cell, but they also have a threshold for length. And that length is, we are able to make that threshold because there is a huge amount of data available in the literature to support that decision making. This is not true for many other decision nodes in many other IATAs. So it's almost impossible to have that level of certainty to make these kinds of thresholds for many of these other decision-making processes. In the interest of time, I'm just going to move on to talk about how we can make these decisions. So I'm going to move on to the similarity assessment. So instead of using these thresholds, there are algorithms that we can use to assess how similar nanoforms are. So are they sufficiently similar to each other to be grouped? And are they sufficiently similar to each other to allow you to read across or copy data from one nanoform to the other in order to fill data gaps? So the REACH regulation from ECHA says that you can do this, providing that you can justify your similarity. In fact, similarity is mentioned 23 times in the ECHA guidance on grouping and read across, but there are no methods proposed as to how to assess similarity. We are writing a white paper, which is going to be submitted for publication in the next few weeks into Nano Impact, and will be the lead paper in a special issue which will be dedicated to describing a range of methods which we've developed to assess similarity. So often as toxicologists or people doing risk assessment, we generate a lot of data. And a lot of these data come as distributions of data. So it could be size distributions for particles, it could be dose response curves for toxicology, it could be ranges in surface charge, it could be ranges in dissolution rate of your substance. And it's very time consuming and complicated to compare the similarity of huge data sets of this type. And so what we do, and this is a very common thing to be done in a regulatory setting, is to use something called a scalar descriptor. So a single number that can be used to represent that data set. So you may have heard of the terms no observed effect concentrations. This is a concentration of a chemical where no toxic effect is observed. Above it, you start to see toxicity, and below it, there's no toxicity. Or an EC50, this is a concentration that's toxic to 50% of the cells in a culture, for example. You can use these kinds of values as representatives or scalar descriptors of your data set. So to do that, that allows you then to make these comparisons. And there are various ways that you can compare the data from your data matrix. So if you think about the data matrix generated by the IATAs, it contains lots of data for lots of different decision nodes. So do you do a similarity assessment for all decision nodes simultaneously in a multi-dimensional way, or do you assess them on a property by property basis? So first of all, I'm going to assess these multi-dimensional methods or that allow you to compare all of the different endpoints in one analysis together. These can be conducted on their own or with an unsupervised statistical method, such as a cluster analysis. Cluster analysis is frequently used, or these types of methods are frequently used in biology and toxicology now to represent patterns in data. So there are two ways of presenting this kind of data here. So we have a triangular matrix on the left-hand side, and each of these boxes in the triangular matrix is a comparison between two different nanoforms. So these are names of nanoforms on this vertical axis and this horizontal axis. So this is a comparison of a nanoform with itself here. So green shows that they have a high level of similarity, and red shows that they are completely dissimilar to each other. So there's no similarity between them. So comparing itself to itself, it's high level of similarity.
So in this diagram here, you can see quite a few green boxes showing that these substances are similar to each other, and then a few orange ones showing that these substances tend to be quite different from the ones along this part of the axis here. Another way to present it is with these cluster diagrams, where you can start to pick out where you have similarities and how these similarities grow. Unfortunately, we have compared about 4 different methods for doing this. It works, you can do it, but you get quite different results between different algorithms, and we don't have enough evidence yet to say which of those is the best one to do. They demonstrate quite interesting patterns in the data, but it's not sufficiently robust to support regulatory decision making. So it's more of a research tool at this stage rather than supporting regulatory decision making. Whereas the pairwise property by property analysis are much more robust in terms of the decisions that you can make. So here are 4 different pairwise property by property analysis. So here we've just got cytotoxicity, or toxicity to the cells in vitro measured, and similarity assessed by 4 different methods. Here is an X-fold difference between substance A and substance B. Here's a Bayes factor analysis, here an OA analysis, and here an absolute difference analysis. Again, green means they're highly similar, and red means they're not similar. In the top of each diagram, it's exactly the same data set in each one. These are our benchmark materials, our positive controls and negative controls, and then these are substances, similar substances compared. So you can see it's picking out the similar substances as being similar to each other, and it's picking out the benchmarks as being different. So it's working well. So they all seem to work quite well, but the one that's least convincing is the absolute difference one, and you definitely need to conduct some kind of transformation of the data, such as a YJ transformation of that data, to make it suitable for this purpose. I'm conscious of time, so I'm just going to move on to my concluding slides because of the technical issues that we've had. And just to say that we're undergoing a number of case studies at the moment, internally and externally, with research partners, with consultants that represent industry, and with policymakers, to test the framework, the IATAs, and the similarity methodologies to make sure they're suitable for work for using. And we are making this both as a guidance document, so a document that takes you step by step through the process, but also as a software blueprint that can be incorporated into other risk assessment software tools in the future. And I'd like to finish by thanking the whole of the Gracious Consortium for all of their hard work in putting this together, and I apologise for the technical difficulties.
Thank you so much, Vicki, and no problem at all about technical difficulties. These things happen, eh? So I'd just like to open up the floor for any questions. Maybe I'll get the ball rolling and ask one of my own. I think we have around 10 minutes that we can use. So I'm really impressed. I think the framework looks great. It kind of reminds me of a Cambridge Engineering selector, but even more comprehensive. It's amazing what you've managed to do in such a short space of time. I'm just wondering whether, from it being so comprehensive and covering such a wide breadth of kind of nanoforms, what were some of the main challenges that your team had to overcome in creating a framework that can be used for so many different contexts?
So creating the IATAs was a huge amount of work. We went through several different iterations of making those IATAs before we could get consensus on how to clearly present them and what was the most evidence-based and logical way of presenting them. So that was a huge amount of work to do that. The next thing, which I thought would be more difficult, was the similarity assessment, but for some reason it was something that really grabbed a lot of people's attention. And so what we thought was going to be a small working group to develop these methods was something that everybody wanted to get involved in. So suddenly we had 30 people on that working group. So it turned out to be, we had so many ideas to choose from that we were kind of spoilt for a choice. So that was quite a nice positive outcome for something we thought was going to be quite difficult.
Oh, Francis, you're muted.
Sorry, I said thanks. Yeah, that it just seems incredibly useful. I'm amazed with what you've done. Does anyone else have any questions from the audience? Feel free to unmute your microphone.
Yeah, I can go first.
Oh, sorry,
Yeah.
I saw the raised hands. Yeah, go ahead, Adolphus.
Thank you so much, Francis. Good afternoon, Professor Stone. Thank you so much for your graciousness in sharing the gracious framework, and I really enjoyed the content. Okay, I just have like 2 short questions. I will just go one by one. The first one is that I'm just a bit curious whether this framework that we just talked about could be extended towards online learning, integrative online learning, in the sense that, you know, when we have ever-changing information of the material with time, you know, is it possible where that this online learning could be integrated within, you know, the framework itself?
I don't know the answer to that. I'm not an expert in that area. I think, you know, using artificial intelligence is something that would be a really great step forward. The partners who have developed the blueprint are called ThinkWorks. They're a Dutch company based in the Netherlands, and they have been amazing. The guy, Ralph van Houten, who does this, he understands the whole project. He understands every single bit and has worked really incredibly hard to make this blueprint. And I think if there would be an intelligent way of updating this with time, that would be amazing.
Yeah, because I thought that was actually because what we presented was more of a like offline static kind of thing. But if something that's dynamic, especially when you integrate this with, let's say, in a medical context, you know, with biological setup, that would definitely be like something that is applicable.
Yeah. But, you know, one of the things that about 10 years ago, I was involved in a number of other projects where we were trying to make frameworks to support risk assessment process for nanomaterials. And, you know, there was always this kind of desire or push to try and make it accessible for non-experts. That's a nonsense. We are never going to make these systems, these methods, these tools accessible to non-experts. It's just too complicated. You can't simplify it enough to make it suitable for that. It's always going to require some expert judgment, and that's why I pointed out the input of consultants at the beginning and during the case studies, because these people are really going to have to support industry in using these kinds of frameworks. And so, you know, to build that level of expert judgment or complexity into an AI could be quite tricky.
Understood. Yeah. Yeah, thank you so much for your views on this. My next question could potentially be something related to what Francis just asked, and I think you might have answered it. But I was just wondering, do you foresee, like, you know, any potential drawback in this framework, like, you know, in this application any context?
Drawbacks is that, you know, it's not accessible to the lay person. Definitely not. You do need some expert judgment in there. That would probably be the biggest thing. And I think there still is quite a high level of information requirement. I mean, we've trimmed it down, streamlined it as much as possible, but working with the consultants, they would like to have a situation where you could just have a single liquid media that they assess dissolution in. But unfortunately, the body is not that simple. The environment's not that simple. There is no one system where you could assess dissolution that would be representative of what's going to happen in the lungs, gastrointestinal tract, the skin, the aquatic environment. So there still is quite a high information requirement.
Understood. So it's really like limited to a big data kind of like setup for sure.
Yeah, but there are databases available to support this, and these databases are expanding with time. There was a project called eNanomapper several years ago, which created a database, and that database continues to be expanded by projects like Gracious and other projects by feeding in data as we generate it, and it's open access.
Awesome. Right. Thank you so much once again for your input and your views on and addressing the questions that we have. Thank you so much, Professor Stone.
Thank you.
Alex Wimbush, would you like to ask the next question?
Yeah, thanks very much for the presentation. Cool to see, always cool to hear about nanotechnology and things. I was just wondering, could you could you run over the process of refining the hypotheses? Like there was the stage of you generate the hypotheses for these material, and then if it fails, you either have to make the decision to reject it outright there if there's no possible means of refining, or you can refine it and then retest. Is that second test like dealt with independently of the first one, or is there some consideration of the fact that this thing will have gone through two possibly related hypothesis tests?
Yeah, so I think when you refine the hypothesis, you're not starting from scratch.
Yeah.
You've got this huge data matrix of information, and probably what will happen is you've got all of these questions, these decision nodes, and you will see similarity for some of them, but you won't see similarity for all of them. And so you know where your hypothesis stands up and you know where your hypothesis doesn't. So it allows you to refine it in an evidence-based way, so you're not just guessing. Does that help?
Yeah. Would that information not be used in if you've got the information there for the similarity along these different characteristics, would that not go into the initial hypothesis test? Like what does the refinement step introduce?
Yeah, so I mean, you actually raised a really good question, because if you don't see similarity, you've got kind of two choices. You either reject your hypothesis, and actually what we often see is we might have, say, 10 nanoforms, which we've got in our initial group, but we accept the hypothesis for 7 of those nanoforms, and we only reject it for 3 of them. So what we're saying is we can group this 7 nanoforms, but we can't group those 3. And that's what we're tending to see is the most sort of common outcome from the case studies that we're doing. But in actual case, we haven't, I suppose the refinement has been part of our research process. So the hypotheses that you see there today that I presented to you and that are going to be published in it have gone through that kind of refinement step over and over again to make sure they're really clear, to make sure that they have the information in there to allow you to logically group them. But I think the refinement is something that has been more of the research process. I don't know if it will end up being part of the user experience, but I think it will for some people, not for others.
Okay, yeah, that makes sense. It's like the refinement is a sort of like a call for more active research investigation into the similarity that you're interested in. If there is a belief that maybe there is something there, but it needs some more work to.
Yeah, exactly.
Yeah. Okay.
Yeah. But you might decide that's too much work to come up with that evidence to do that, and that, you know, maybe you should just do your risk assessment on a case-by-case basis. It's a balance.
Yeah. Okay, thank you very much.
I'm curious, Vicki, what is some of the kind of worst case kind of nanomaterial risks that you've seen or encountered where you've kind of come across maybe a sort of nanotechnology that we should definitely avoid? What are some of the worst case scenarios that you've encountered?
I mean, it depends upon if you're talking about human health or the environment. And from a human health perspective, you know, one of the multi-walled carbon nanotubes, Mitsui 7, has now been classified as a possible carcinogen. It seems to, you know, these long, fire-persistent, rigid nanoforms seem to behave like asbestos. And so you wouldn't want to use them in any applications where they're going to be aerosolized and inhaled into the respiratory system. And I think there's kind of wide acceptance of that. Obviously, there's some naivety with some developers. People don't always understand that. But yeah, there's wide understanding, at least in the regulatory and research perspective, that that's an issue, and more so in industry. Then we have things like quantum dots, which have cadmium in them. I mean, why did somebody invent a quantum dot with cadmium in the core to inject in the body for imaging? I mean, you just need to ask a second-year toxicology student, and they would have told you that was a bad idea. But you know, they're research, they're a step towards getting other safer materials that could be used for imaging, but they were never going to be used in the body. something that was quite an interesting conversation the other day. Zinc oxide, you think is quite an innocuous, low toxicity substance, but actually in the environment, even with a low dissolution rate, can release enough zinc to be significantly toxic to organisms in the environment, which I was quite surprised by. So there's all kinds of examples, but there are also an equally high number of examples where there's no measurable toxicity or no significant effects that you can measure. There's the full spectrum.
Okay, thank you very much. Well, let me thank you again on behalf of everyone here and Scott. And yeah, I wish I could offer you a round of applause in real life, but obviously, hopefully we can maybe see one of your presentations in person one day. So thank you very much. So let's move on to our next speaker, Igor Linkov. So Dr. Igor Linkov will be presenting his personal reflection on 20 years of nano risk assessment and relevance to emerging risks. He's a senior science and technology manager with the US Army Engineer Research and Development Center and adjunct professor with Carnegie Mellon University. He's also responsible for the ERDC's project portfolio in areas of crises management and resilience. He's developed methods and tools for measuring resilience in interconnected network and applies these tools to critical infrastructure, transportation, energy and cyber systems, supply chains, and also currently to manage the response and recovery following the COVID pandemic, which is really interesting. He is Army representative at the National Nanotechnology Initiative, Networking and Information Technology Research and Development Program, and he's published very widely on environmental and technology policy, climate change, and risk and resilience analytics, including 25 books and over 400 peer-reviewed papers and book chapters in top journals like Nature and Nature Nanotechnology. So I'd like to welcome Igor, and if you'd like to start sharing your slides.
So you don't see my screen, do you?
No, not yet.
Why don't you share slides again? I'm using somebody else's computer and it's Apple. I'm not familiar with them.
Just bear with me and just let me know when you want me to change slide.
Sure.
I'm just making sure I get the right perspective first, because I need to close the existing slides. Can you see this okay?
Yes.
Is that the right view?
Yeah.
Okay, perfect.
Okay, well, thank you very much. It's always difficult to follow Vicki. She's such a great presenter. And I'm really was pleased to hear about recent development in Gracious. It's a really important project. But let me ask you a very simple question, just to start thinking. What would you do if somebody asked you to compare a similarity of dogs and cats, right? Obviously the question is, what perspective are we taking? If you are a zoologist, you will find that they're completely different. And, you will find cluster analysis of, God knows how many ways to show how different they are. But if you're looking for a pet, it may be not so different, right? So the message here is that somehow as scientists, when we try to do something that has policy implication, we believe that we know what decision makers needs. And we often develop very sophisticated constructs, like the one that we saw, that kind of answer questions that we may, as a scientist, or may not capture what decision makers really need. And in my presentation, I will try to show some of the reflections on that, how judgment that we make as scientists may be off and not consistent with what really needs to be done from decision makers' point of view. And of course, in the time of crisis, like COVID crisis, it's like perfect situation where scientists need to provide support in the area that they have very little comfort, or often very little comfort and knowledge. And nevertheless, we need to provide advice. And I would argue that early stages of technology development are very similar to crisis situation. Anyway, so let me start my presentation. So next slide. So I am from Army Engineer Research and Development Center, and we are a large organization. We have some 2,100 people, have PhDs and master level. So our mission is to do science in support of Army Corps, Army and government in general. But you know, I always need to, especially in Europe, need to say that we are really not military. You know, most of the research in the US, not most, but significant portion of the research is done by Department of Defense. Our budgets for research are, much, much higher than National Science Foundation and all other civilian agency together. So it's inevitable that in many areas, including emerging technologies, military conduct research of societal relevance, and Army Corps in general, it has both civilian and military mission. So on military side, we really look at advanced technologies, including nanotechnology in this case, but we have a civilian space where we look at environmental management, energy, recreation, navigation, things like that. So my team is in Boston, Massachusetts. Organization has 7 labs. My lab is in Vicksburg, Mississippi. I was recently promoted to become kind of portfolio manager, so I'm working across the organization now. But next slide. What I'm trying to do, I'm trying to integrate physical and social science. And risk assessment, of course, was one area that I was engaged from my very early days in practice. And Vicki already started to address the issues related to deficiency and risk. Risk is product of threat, vulnerability, and consequences. And especially for emerging materials like nano, it's very difficult to find all components that we need to do risk assessment. Uncertainty is just way too high. Next slide. Before. So, and actually I was working on nanotechnology in Venice and back in like 2013. And this is where I kind of start to develop interest in pandemics. And of course, Venice is associated with plague. Some 700 years ago, we had a really important pandemic. It's originated, in China as well. And in the time of 20 years, it reached Venice. Next slide. And you can see here that in another couple of decades, 2/3 of population of Europe was dead. So with COVID, when people were saying this is unprecedented, it actually was much better than what we had in Venice 700 years ago. And back to risk assessment, next slide. Probably you can click a couple of times, it has this animation. One more, yeah, stop. So when I was in Venice back then, I was thinking like, okay, if I were there 700 years ago, what would I be able to do as risk assessor, given state of knowledge we had back then? And obviously, people thought that plague is coming from God, all right? It's and the way they mitigated, they were building all these beautiful churches that are still almost the same as 700 years ago. From medicine point of view, belief was that, well, you have skin deformations. So what they did, they apply metals. Obviously, 300 years later, we realized that that was not going to help. And the reason for plague was very different from coming from skin. And what was very interesting is that they really believed that vampires were spreading the disease. So how you deal with vampires? Well, you just put brick in mouths of dead people so they don't chew their way out of graves and don't spread the disease. So this is like all a long way to say that probably risk assessment as methodology would not be very relevant to decision makers in Venice at that point. And next slide. Next slide shows actually, little did I know back then that I would be in a situation of, thinking what to do with bricks and how to put them in the mouths of dead people and things like that. Like late March in 2020, when COVID crisis was just starting, I was contacted by our Pacific Ocean Command, and they are in Guam. Guam is very far from US. And they have significant military presence. And what was happening in Guam from with COVID, governor of Guam was saying, well, we over, crisis is gone, we flattened the curve. That was her words. And the reason for that was like a profile of cases. You can see like perfect peak, the decline, and the late March, they have only like whatever one case. And at that point, governor was saying, well, yeah, we are done. We don't need any military help. And military was kind of uncertain, what should be done, because Guam is far. To be able to bring materials and supplies and treat people, they need like advance notice. So they basically call us and say, well, can you do modeling to show progression of the disease in Guam so we can talk with governor about it. So I will leave you with these thoughts for now, but that was like a real situation Friday evening, and everything needs to be done by Monday. They need to talk with governor. And yeah, what would you do as risk assessment? But anyway, next slide. Let me leave you with that thought and go through presentation. So I will start, I try to do very complicated things, so I'm not sure how it will work, but I try to blend my personal history of kind of maturation as a scientist and engagement with nano and talk about judgment and modeling related to nano risk assessment and governance, what we are talking about. And I like to finish with the ideas of resilience that is actually what I'm currently working on. But next slide. Let me start in 2004. This is where I first started working on nanotechnology. I'm not sure when Vicki started, probably about the same time or maybe even earlier. But this is from our paper when we tried to kind of parallel nanotechnology and synthetic biology. And left side shows carbon nanotubes, a number of papers in physical and social science. We tried to cluster these papers into groups. And you can see that carbon nanotube, which was one of the most developed materials at that point and still is, I guess, we had already in 2004, 20 years of technology development. We had some 10,000 papers published on nanomaterial technology. And on social side, on environmental, legal, and social implication of a carbon nanotube, we were literally in double digits, probably 70 or 80 papers. So that was kind of early stages of the field. Of course, at that point, I didn't realize how little did we know at that point, but you can see only like 70 papers overall in environmental side of things for carbon nanotube. And just a side note, because I will mention SynBio, it's probably where we are now in SynBio. The difference is that if with nano we had 20 years of technology development before we start asking questions of environmental and social and legal side, in SynBio, the time lag is only like 2 years. But anyway, so we are in early stages of the field. Environmental side is only getting popular, so to say. Next slide. This is what we thought about nanotechnology. We were all excited. It was all kinds of prediction that by 2015 we'll have, 3 trillion market for nano, 15% of all manufacturing will be nano-enabled. Obviously, all these predictions didn't come to fruition. We were off by literally order of magnitude, but it gives you kind of spirit of the time. Next slide. And by the way, I'm using a slide unchanged from my presentation of 2005. So just to show, my point here will be how little that we are still asking the same question some whatever 17, almost 20 years after. Back in 2005, Environmental Protection Agency decided to draft the first nanotechnology white paper, and they convened a panel to evaluate it. Next slide. And somehow I was invited to be part of that panel along with Andrew Meinert, Dan Tamalia, who was one of the top developers in the field of nano, Richard Dennison and others. So I was very young. I don't know how was I selected at that point, but anyway, next slide. The panel was evaluating state of the risk assessment for nanotechnology. And again, this is my slide of 2005. I did a couple of presentations based on this panel recommendation. And you can see that the same questions that kind of parallel to what Vicki was saying. We have same regulatory challenges, and from risk assessment, we still have deficiency and inability to do risk assessment the way it was done for chemicals. Next slide, quickly go through that. So that was our recommendation, that we basically need to develop grouping systems for nanomaterials and frameworks through the life cycle. It's almost like, if you kind of go back to Vicki's presentation, it's almost the same questions. Next slide. And I guess at that point, I was really adding, I remember how I started this. I was adding this kind of decision analytics to this, because at that point, even though I was very young, I had already some 10 years history of applying risk assessment for chemicals and radionuclides in the environment. And I kind of felt that uncertainty will be very high, and you really need to do integration of physical and social science. Next slide. And background, my personal background for that was this work that I did for my PhD dissertation. I modeled radionuclide migration in forests and fruits. And about like in late 90s, early 2000, we went through exercises with International Atomic Energy Agency when they spray strawberries and other plants with radionuclides, measure what happened, and ask modelers to blindly predict some of this and compare what we can predict even for a very simple system. And next slide. Next slide shows some of the results. So what happened is they spray radionuclides, and we have like model runs, like every 6 months we were meeting and we were comparing results, right? So what happened is first time we met, we have like very significant difference in model prediction. And this is all like different models by different agencies in Europe. And my model was part of that. And then after several meetings, the difference in model prediction kind of getting smaller. And eventually we have, in this case, something like 1 order of magnitude difference. So in this case, interpretation was that model uncertainty can be as high as a couple of orders of magnitude. But this difference, if you do probabilistic analysis, parameters uncertainty may contribute 1 order of magnitude, right? Next slide. And what does it mean for nanotechnology? I think many algorithms that Vicki was presenting is actually driven by QSARs when you try to kind of cluster properties of nanomaterials together. So, so what can you get based on difference in different models of QSARs? So this was actually a paper that we did back in 2007 when I just joined Army Corps of Engineers, and I was asked, okay, how much uncertainty are we getting with QSAR prediction for explosives? And what we've done, we put same explosives in 6 different commercially available software tools for QSAR analysis. And next slide. Click once more. So next, so this is shows pesticides. You can see that all QSAR model can predict pesticide really well, but click one more. When you put in these QSAR models explosives, all of a sudden you get this couple of orders of magnitude uncertainty in prediction of KOW, which is like constant for a chemical. And it shows like uncertainty in all this clustering algorithm that we used, in this case QSAR, but it's probably not too far from other clustering tools that Vicki presented just a minute ago. So next slide. Next slide shows what is even more worrisome based on predictions or exercises that we run with International Atomic Energy Agency. It's what I call modular uncertainty. And the problem is that when we try to predict uncalibrated scenario that doesn't have calibration point, something that really unknown that we didn't experience before, all of a sudden we just cannot predict. We, as a scientist, when you see something not familiar, you try to benchmark based on your understanding of the problem. And as a scientist, you are quite confident that, you know, whatever the way I see the problem is the truth, right? But what I show here is that, you know, if you model forest with acute deposition of radionuclides, something that we all experienced in Chernobyl, it was like, you know, hundreds of millions of dollars spent on modeling acute deposition and calibration of all the models. All of a sudden you go from like 1 to orders of magnitude of model and parameter uncertainty to literally up to dozen orders of magnitude, just because modeler's interpretation of scenario and what can happen may be very different from one to another. So, and this is like true fact. It's not like in these exercises, if we had questions, we can go to the agency, they would clarify. But it's overconfidence of modelers. It's well-known fact in modeling literature, but it shows magnitude of that. So next slide. So remember my problem with Guam. So the question, I was asked to predict what's going to happen in Guam with disease spread. And at that point, it was very little information, right? So what would you do? And basically what we try to do here, and it shows, we try to answer the question. The question was really not what is going to happen with Guam, but rather, should military be worried? Should governor be worried? And should they request help? And what we try to show, we run all kinds of models that we were able to develop over the weekend based on whatever data was available. It's different SEER model, and you know, basically, eventually our model became part of CDC ensemble forecast, and they kind of did ensemble forecast that they used for prediction. But this is kind of mini ensemble. We just use different parameters, different calibration, and we show no matter what we do, the problem is, it can only go worse. And this was a very clear message that we deliver over the weekend to decision makers. And they, based on that, they immediately changed what they did. Governor of Guam created commission, and they, you know, basically took our model and approaches and develop, you know, more of that and make recommendation to, well, eventually we went to lockdown. But next slide shows what's actually happened in Guam. So around late March, we had this local minima when we get engaged. But on the bottom, you can see the actual curve. They indeed had a peak in April, but then the second wave hit them much harder. So anyway, the message is that subjective issues in the modeling and in crisis response and in new technologies is very important. And it requires integration of physical and social science tools. Next slide. So what can we do? And this was kind of line of work, and I work in Nano from 2004 till now, actively engaged in as a part of National Nanotechnology Initiative, and supported some European project. So next slide. The core problem, and again, this is what I argue from the very beginning of my engagement, is that as long as you have emerging technology that evolved much faster than our ability to generate data about this technology, these gaps between in risk data and ability of regulators to analyze this data and develop guidance is only goes to broaden, right? No matter what methodology and tools we develop, we will have less ability to guide technology just because data is still not there. Next slide. And as I mentioned, back then, I was arguing for use of multi-criteria decision analysis that explicitly acknowledged that you just cannot have unbiased, completely, physical or statistically driven approaches and tools. You need to integrate judgment into the process, both regulatory and expert judgment. And rather than try to hide behind sophisticated statistical algorithm, you just need to acknowledge explicitly that there is a judgment, there is data, and they need to be combined. And next slide shows methodology that we propose for grouping of nanomaterials back literally 15 years ago. It requires selection of a few properties of materials that can be measured really well, and making decision based on this grouping decision based on these properties. So, and this grouping is driven by two factors. One is performance of these materials based on these properties. Some of them might be more reactive, agglomerate faster, be smaller, or be of more aggressive shapes. But then there are no models in the world, at least at that point, there were no models that would really connect shape of carbon nanotube in this case, or other materials with the hazard and risk. So instead of just try to model that, you can just say, well, we believe that shape is more important than whatever number of critical function groups. So you kind of, instead of doing model, you integrate your judgment as a decision maker, what is important for you. And that can be used as a reason for grouping, and that papers conceptually show that it's possible to do. Next slide. I will come back to this because that methodology was actually, we developed it further, and it's now part of how Consumer Product Safety Commission is grouping nanomaterial product, nano-enabled product for their testing. And it has been in practice for last few years, and now we're working on expanding that to include multiple chemicals, not just nanomaterials. So obviously, very rigorous methodology that Vicki presented is significant development in the field, and it's well thought through, but still the question is, do you really need anything like as sophisticated as that to answer regulators' question? And I would argue that in case of Consumer Product Safety Commission, it's not necessary. For screening level assessment, for many other problems, you can do much easier and less sophisticated evaluation and still be helping regulators. And this is kind of bringing me to the whole idea that the field of nano kind of gravitated towards. So initially, first, whatever 10 years of nanomaterials research from early 2000 to early 2010s was try to really improve risk assessment, and eventually we stopped and start to move to alternative ways of doing that, and Vicki presented groupings, but we kind of embracing a broader set of tools for that under risk governance. And risk governance tries to use proxies for risk assessment through different methods and tools, including cost-benefit analysis and risk evaluations, not necessarily assessment, maybe even with judgment, and driven by risk management. So I don't have time to go through that, but next slide. Next slide really shows some of our work that if a lot of governance, a lot of risk assessment or risk-based governance is hard law, like this is how EPA and FDA regulate many things. There is also soft law that more like close to risk governance side. It's codes of conduct, public-private partnership, multi-stakeholder engagement, and things like that. So governance needs to be based on both of these. Next slide. And there are other methods and tools that evaluate, including cost-benefit analysis, control banding, decision analytics, adaptive management, and things like that. Next slide. And fundamentally, what we argue that traditional risk assessment starts with kind of bottom-up approach. It starts with data and try to capture the data through mechanistic model and statistical models, and then go to decision. You basically start with data and you visualize and try to provide interpretation of the data to decision makers. And next slide. And in our own work, we've done what we call Nanogrid, which is kind of decision platform that allows to do risk-based decision making, starting with data. Again, Vicki kind of went through that extensively, so I will not go, you know, everybody has different tiers of the analysis and all that. It's kind of a feature of many bottom-up methods. You do through series of rigorous analysis, data-driven rigorous analysis to really reach decision point for decision maker, right? And you build all this logic model. If this tier tells you X and you know you have a Y, then you go through direction Z, right? So those are very data consuming, and indeed they may be very valuable in some application. But next slide shows that what I was talking about before is decision analytics tools that actually start with decision maker values and needs, and then through decision modeling, they frame the problem so that we are not looking at the all volume of evidence-based data, but rather we focus on a few factors that are relevant to decision maker. And by doing that, in situation of high uncertainty, you can remove a lot of data analytics that may not be necessary. It may just add noise to decision makers' ability to make decisions, right? So next slide. And this is kind of framing that we used, and we published it in multiple papers. And I guess next slide. So basically we go from risk expressed as product of threat, vulnerability, consequences to metrics of risk, metrics associated with threat, metrics associated with vulnerability, and metrics associated with consequences or hazard exposure effects and kind of toxicology framing. And this is exactly what we implemented for Consumer Product Safety Commission. This was published in Environmental Science Nano a couple of years ago. And next slide. Next slide shows how we really look at the properties of nanomaterials. And what makes it easier to do is we are not asking questions of what is the risk expressed as whatever cancer risk or other kind of absolute risk value that needs to be compared to threshold, but rather what we do here, we look at nano products, and we try to say, well, these products are most risky than those products, right? So it's very different framing of the problem. You go from absolute risk to comparative risk assessment, and it's eliminate and needs to do a lot of quantification that it's really important on the REACH framework and on the other frameworks that, you know, actually Vicki was presenting. So next slide. So yeah, I guess we apply this methodology for synthetic biology. So for us, this is what really constitutes risk governance. It's departure from hardwired risk assessment to kind of embracing multiple criteria associated with risk and benefit and societal impact and all that. Next slide. And well, the question is, what kind of data tools that you can bring to help you to do that? Well, you have very complex data intensive tools like AI, computational toxicology and all that. And on the top, you have very simplified processes, just simple ranking and all that. And decision analytics is somewhere in between. Next slide. Next. Yeah, so, and I think now we have, I think 4 projects that look focusing on risk governance for nanomaterials. I think they focus on creating a risk governance council, and I think it was like coordination across all of them. Again, I don't have time to go through that, but this kind of shows the progression of nano risk assessment as the field from, you know, try to really do risk assessment the way we do it for chemicals, now getting to governance side. Next slide. And well, it kind of brings me back to Venice, right? So, and it's very important for the future, and I do believe that will be future of all emerging risk. What we argue is that in Venice, they actually didn't deal with risk. Risk was high, and you cannot deal much, you cannot do much with risk if you experience this for the first time or experience some variants of that like COVID for the first time. And remember with COVID, we had all kinds of models 1 year ago predicting a very different outcome. And we still, you know, we support, after this Guam things, my team basically provided most of decision analytics work for 6 states in New England, and we still work for Health and Human Services and FEMA. And the question is what's going to happen in the fall. It's very high on our agenda now, and there are no models in the world that can predict what is going to happen. But again, what I'm trying to tell them is that the question is not to predict how many cases we'll have, but rather, should we be ready? Should we have resources in place? Should we be able to support, if you are FEMA or HHS, should we be able to stockpile resources to help with pandemics if we have another wave? And I think from point of view of predicting that it will be another wave next fall, most of the epidemiologists are consistently saying that it's very likely to happen again. So what they've done in Venice, actually, they look at resilience in our today's terminology. They were the first to use protective clothes. They sent people to Lazaretto Islands, and they quarantine ships that come into Venice. In this, on the right, on the right, bottom right, you have Lazaretto quarantine island that is still exists today, and some of these remnants of structures. And what does it mean? Next slide. It means that they try to break disease propagation network at individual levels through protective clothes, at local levels through Lazaretto Islands, and at regional levels through quarantine. So they break a network of disease propagation. And next slide, it really means that they try to implement resilience features. And inspired by my Venetians ideas, on the tail end of Ebola pandemics, we developed this paper when we model pandemic spread, and we try to argue, we try to study what a pandemic, how you deal with pandemics from point of view of risk and resilience. And next slide shows kind of our results. And this is pre-COVID. What we showed that from risk point of view, travel restriction, and in the paper we also look at fear, how you embed fear in population. From risk point of view, more restriction you have, less risk. So no matter how strong, how weak is pandemic, it's beneficial to keep people at home and scare them to death. But from resilience point of view, more you do that, it's more difficult for you to recover. And there is this narrow range of disease pandemic size that would justify restrictions and before you kind of get in these regions where it doesn't make any sense. And unfortunately, this was not done for COVID. Methods and tools that embed risk and resilience were not developed. And yeah, we were basically in dark when we try to help decision makers to deal with this. Next slide. So what we argue actually, we need in new technologies in crisis, we need to accept this unknown. We need to accept risk, and we need to focus on ability to recover. Because again, from a decision maker point of view, if you may have high risk, but if you can recover from failures of that product or that system, it may be as good as having low risk, right? As long as you can do that. And many of our systems are designed to be robust so they don't fail. And this is the whole field of resilience analytics is focused on doing that. And we argue, especially for synthetic biology, this is kind of from our SynBio paper, we argue for SynBio, which kind of technology on its own that kind of evolves, these ideas of resilience needs to be embedded for sure. Next slide. Well, I guess that's pretty much my main message. And what I want to say now, back to the field of nano, social and physical science within nano are really embedded. Again, this is kind of from our paper on SynBio when we try to show that it's natural progression of this high emerging risk materials that eventually social science and stakeholders and public perspective on that is start to drive physical science. And that's what happened with nano. We're now talking about safety by design, and hopefully for other materials it will be the same. We can go much faster through this period of trying to do risk assessment and then getting to social science side of that. Next slide. So combination of this bottom-up and top-down approaches can really help in making this transition much faster. Next slide. And it's very important to look at value chain that allows to kind of connect developers of new technology and operators and users together in kind of holistic approach. This is where value of resilience is going to materialize. Next slide. And globally, again, in the field of resilience, we have this idea of panarchy, that society needs to go through cycles when they have high production stage, and then you have crises that allows them to go back. So after Black Death, we had period of Renaissance when population become healthier and produce more and develop all these arts. So back in 2016 in this paper, we argued that we need to be smarter. We need to go to the next level of societal development without collapsing. And I hope that that will be the case with COVID. When we wrote these papers, we didn't even expect to have the situation that we are observing now, but hopefully a civilization will be able to make right lessons out of that. Next slide. And yeah, we published a lot of papers, and next slide shows some books, including the new books that we try to use on COVID systemic risk and resilience. So I will be glad to answer any questions. Over.
Thank you very much, Igor, for your really wide and diverse talk. It's clear that you've collated a lot of your own work and reflections. It's nice that you kind of cross-examine different areas. Has anyone got any questions from the audience? Did Alex raise his hand, or?
Yeah, I was putting the hand down. It was still up from before. Sorry.
From your experience, Igor, with regards to kind of nanotechnologies and nanomaterials, I know you've basically showed that in the past there was a bit of a kind of hype bubble with regards to the expectations of the industry is going to grow massively into these billions and billions of dollars. How much do you think we've missed the mark by, and how long do you expect before it becomes more mainstream essentially and it's used in almost every application?
Well, I'm not sure. I think there is a really big value in the type of work that Vicki is doing and others, because again, this is kind of hard law. It's part of REACH-based regulation, and these types of laws and approaches allow us to really clean up environments substantially and don't have major failures of new technologies. So all I'm saying is that at some point we need to stop. At some point, once you kind of prevent the obvious issues related to new technologies, you need to allow industry to develop. And this is where some of these resilience-based approaches come into the play. And we just need to do more on that. Over.
Thank you very much. So does anyone have any other questions in the audience? Oh, Conal, if you'd like to unmute yourself.
Hi, you said that you're involving more and more methods that involve judgment rather than data. How do you reach an agreement on what the judgment should be? So how do you reach a consensus on value judgments in these analyses?
Yeah, so you're absolutely right. It's very difficult to reach value judgment consensus, but many decisions are done by individual decision makers, so it helps in many cases. In other cases, you can do scenario analysis. You can really show that for a wide range of values, one technology or one configuration may be preferential to others. So yeah, that's kind of approach I'm taking. Rather than try to reach consensus on exact numbers, I'm trying to show kind of this exploratory space. And after all, in many of these decision analytics tools are designed not to tell you what is the best for you, but rather stimulate your thinking and allow you to explore space of decision space to really be able to do that.
Okay, thank you.
And I believe we have a question from Professor Vicki Stone. If you'd like to unmute yourself.
Yeah, thanks. It's a comment and a question. So the comment is that I'm an academic. I don't work for a government organization. So the research that I presented to you today is to help society work within the regulations, the governance structures that have been set by European regulators, and which has now been adopted into British regulation as well with Brexit. Whereas Igor is in a position where he, in some of the work that he presented today, he has the opportunity to influence policy decision making. But obviously that has a political angle to it. So I mean, Igor, I don't know how much you can talk about politics, if you're allowed to or not, but there's a big political change in the US now, but when COVID first started, it didn't matter how much evidence you had, how much evidence were you able to use, how much science were you able to use to influence decision making?
It's actually not necessarily the true. statement about like previous administration and science and politics in general. The decision making in the US is very decentralized. So, and it's for the reasons actually to be resilient. So, for example, I worked for regional FEMA and Health and Human Service administrators. And yet they were under political pressure to do some things, but they were authorized to ignore it basically. So I think that's some of the difference, like, between US and Germany to some extent, for example. Regionally and in each state, the governor can do pretty much what he or she believes necessary to do, ignoring or taking to consideration what they believe is important from federal guidance, right? So from my experience, working with regional administrators, we basically provide all data analytics, right? They started like calling me for like a couple of weeks last March and saying, well, we need a statistician to look at the data, to really employ and like on detail, like 4 people from my team doing a full-time data analytics. Till now, we actually on their payrolls through end of June. And so we've been through all crises, and we did pretty much everything from resource allocation in hospitals, return to work, vaccine distribution, you name it, equity issues. We really provide data-driven approaches for that. And it was immediately used by regional administrators. And this is like federal administrators, right? They worked for Trump before they worked for Biden now. So, for military is not political at all, right? So generals are not reporting, well, we have only in military, we have political appointees like Secretary of Army or Secretary of Defense, but the rest of the generals stay no matter what administration is. So I guess this is a long way to say is that appetite for evidence-based data is really high, and many decision makers at the highest level, at local level and federal level, are still driven by evidence rather than judgment. But with that said, it's really important to capture judgment and the value space that they have to address, right? So for example, one example was return to work. So remember last summer, the caseload in the US was really low, and Trump administration was pushing really hard to get people back to work. But our regional administrator asked us to analyze progression of the disease and probability of getting exposed in the workspace. And we were able to show that if you have less than 50% in the office, you have risk that increase linearly, but then if you have more than 50%, it starts to grow exponentially. And that was, we informed him that no matter what, don't allow more than 50% of people in the office space. And that's exactly what he did, right? And he used it as a justification for his course of action. So anyway, I guess this may be longer than you asked me to respond, Vicki, but I just summarizing, I think evidence-based data is the key for policy decision makers, and especially under Biden administration, I think it will be even more appetite for that.
Do you have anything to add, Vicki? I'm not sure whether your mic's muted.
No, that's great. I mean, it's good to know that. I mean, it's quite different, actually, the way the US works in terms of using risk information and feeding risk information into decision making at a regulatory and a policy level is very, very different in the US compared to Europe. And I don't know if that's something you've looked at, but the same information, the same evidence can be used, but it can be used in quite a different way. Doesn't necessarily result in a different outcome, but the process is very different. So it's quite interesting to look at that.
Yeah, and this is unfortunate. I really feel that media didn't do a good job informing us about like right course of action. It's kind of was very politicized in the US especially. But yeah, this is another thing, like, you know, as a scientist, we generate evidence, but then all of a sudden, instead of providing holistic picture, media select buzzwords and, you know, few facts to justify completely opposite point of view, depending on which channel you watch. So yeah, it's frustrating, but I guess I don't know what is a good solution from that.
Thanks, Igor. Have we got any more questions from the audience at all? If not, then let's thank our speaker. So thank you very much again, Igor, for your talk. It was really, really fascinating. And I'd like to change over to our open discussion where we can have a brief conversation about our question. If you can see my screen. So the question that's been proposed is, how useful are alternative models and methods in assessing the risks of nanomaterials? Maybe Vicki, you'd be able to start us off with some of your perspective on this question.
Yeah, so there's a whole range of alternative models that can be used that are available now. So these are usually alternatives to animal testing or alternatives to human testing. And they involve in silico, computer-based modeling, in vitro modeling, with cells in culture, and in chemical assessments as well. The key is to understand the limitation of your model. So it may be that you need several alternative models to give you the same answer that an animal model might be, and the level of certainty may be different for those. So the key is to understand what your models can do and what the levels of certainty are. And if you use these kind of tiered approaches that we mentioned in the Gracious presentation and that Igor alluded to as well, that helps to give you a wider range of alternatives and to try and address those issues of uncertainty.
Thanks. And Igor, do you have any thoughts on that question or anything that Vicki's said?
Yeah, no, I just probably will be repeating, but we definitely need to tailor sophistication of the analysis to decisions that you need to make. And, you know, I kind of agree with Vicki. If you have rich regulation in place, you need to have like very complex and sophisticated machinery of how you do all that. But do you need to do it like in all cases? I don't think so. Should we have some alternative way of doing that? Maybe, but again, you know, like my experience with Consumer Product Safety Commission shows that they decided to do something different for screening, and I don't think they will be able even to do what Vicki shows. It just require far more analytical power and capabilities and funding, and probably they don't even need to do that. So anyway, that's kind of my 2 cents here. Over.
And with regards to the kind of communicating these risks and uncertainty to the people that you'd like to influence or the regulators, obviously they might not necessarily be experts in nanomaterials or nanotechnologies. So what's the kind of sweet spot with regards to how you present that information to them in a digestible way that they can actually act on? If anyone would like to speak to that.
I'm not really sure I understand the question. You know, if you're asking how do you present to regulators if they're not an expert in nanomaterials, is that what you mean?
Yeah,
so,
correct.
So the regulators in Europe have gone out of their way to make sure they understand nanomaterials, and they're very well educated with respect to that. So at the level of the European Chemicals Agency, the European Food Safety Authority, the committee that's responsible for consumer safety. All of them have experts in those organizations that have an understanding of nanomaterial hazards. They engage with a lot of the European-wide projects in these areas. They take part in stakeholder events. They contribute to OECD activities. So they're very well informed. So it's more a case of meeting their expectations rather than making it digestible for them.
Yeah, let me maybe try to use COVID as alternative example. So what happened with COVID? Remember, in early days, we had this, the first really model that show up with some conclusive prediction was the UK model, but then it cost the person who developed that model almost a career. I don't know, Ferguson, I don't know what is he up to, but this is like really interesting for me because as a scientist, you need to have significant courage to report something that you are not really confident, and that's what decision makers expect you to do. But then you kind of opening yourself for criticism, and sometimes it may kill you. Remember that the model, IHME model that actually become widely used by White House, that was especially initially a very bad model. You cannot build statistical model out of nowhere, right? They basically took the curve from China and moved it to the US, and that's how they did it. But anyway, they had the courage to do that and put it in the open domain. And even though that was definitely not the best model of the time, they become the model that, it's still probably the most widely used. We developed our own model, which probably much better. It was calibrated and all that. It was kind of blend of statistical model that IHME is with process model. But we were like, 1 month late. So we were part of CDC ensemble forecast, but the first months, IHME model was all over the news, even though they were not the best one. So back to kind of your questions of communication. Yeah, you need to have a courage, significant degree of courage to really communicate your finding. And not many people, not many scientists are willing to do that.
But Igor, there's a big difference, isn't there, between communicating risk information for nanotechnology versus risk information for COVID. COVID is a very acute, imminent threat to the whole world population, whereas nanotechnology is a slower, lower level burning threat, which might be a significant threat in the longer term, but it's not going to kill hundreds of thousands of people in the next couple of weeks. Plus, that gives us the time, therefore, to investigate the risks associated with nanotechnology. And it gives us, therefore, an evidence base to make decisions on. And whereas with COVID, the best you could do is look back at pre-existing pandemics or epidemics and try and decide whether COVID was going to behave like that with very little information to help you to make that decision. So it's a completely different scenario.
Yeah, no, I don't disagree. I use COVID as extreme case, but many of these issues are still relevant for emerging technologies. Yeah, with Nano, now we generated so much information. It was so massive investment that we can deal with that. But then the question really is it worth spending so much effort, so much money on doing that or not? But if you look at SynBio, it's the same as Nano 15 years ago. You have all these miraculous technologies that are on hold because regulators cannot make decisions. So as a scientist, would you take the risk? And no, of course, nobody will listen to me, but if you are a Nobel Prize winner, very reputable person, would you have a courage to say, well, you society should take this in biotechnology and use it is going to save people from cancer? right? If you invest another whatever 100 millions. It's actually some of this COVID vaccine are kind of synbio CRISPR driven techniques, right? So yeah, I don't know. So Vicki, yes and no. I think you're absolutely right. We have luxury of spending more time. Nobody's going to die tomorrow. But with some of these technologies, we are not doing that just because we worry about regulatory impacts. Should we communicate stronger that we're ready or not? I don't know.
And is it black or white when you're proposing your hypothesis or a new material to try and get regulatory approval? Or how do you present the results? Is it, I know in the past, a lot of risk analysts proposed this kind of traffic light system where there's like a hard no, an amber light where it's kind of in between, or just green if it's definitely a go ahead. Kind of how do you kind of approach, or what's the kind of graphical visualization that you guys use to be able to convince people to green light something?
Yeah, these kind of traffic light systems have not been popular because for making these kind of level of decisions, because you end up giving a red traffic light to some substances when they're actually quite safe for some applications. So they haven't worked out to be that popular. The way that, with the grouping and read across approach gives you quite a useful way of benchmarking your new substance to something that you already work with every day, that you've come to terms with what the types of risks are. So, taking a benchmark substance that you know quite a lot about and then comparing your new substance to it is quite a useful way of getting people to think about it. And what the regulations try to get you to do is compare it to a benchmark, but make sure it's safer than that benchmark. So it should never be more hazardous than that benchmark. It should always be less hazardous or similar, as similar as possible. So that's quite a useful way of doing it, I think, because we all deal with hazardous substances on every day of our life. From the soap we wash our hands with, you don't want to eat that, you don't want to get it in your eyes, that you can use it on your hands and it's quite safe. Drinking alcohol, we know how much we should drink, we know how much we shouldn't drink, but sometimes we choose to ignore that advice. Smoking, so we're used to making decisions, risk decisions, even crossing the roads.
Yeah, definitely, that's a really valid point. Igor, what's your experience of kind of presenting the results? Do you follow a kind of traffic light system, or is it very similar to what Vicki said?
No, well, I'm in different space, so like with COVID, I was in a situation when we really work with top-level decision makers, and basically, once you develop trust, they really just trust you, and they communicate the way we like them to communicate. And it was kind of amazing that, basically, they, in crisis situation, they just don't have a way to have alternative points of view, and they just start to trust you as a scientist. Back to nano, it's like much more complicated story, but with Consumer Product Safety Commission, that's the only experience I have in kind of making something like practical with nano as far as regulation comes into the play. They just, for screening level assessment, again, usually screening is done using significant degree of judgment, but so they then, whatever we develop, they use it and trust it. We didn't get in this more detailed evaluation with them. They do it themselves.
Igor, in the early stages of COVID, did you do anything like comparing to influenza or SARS or bird flu? Did you use those kinds of comparisons to try and, benchmark what COVID might or might not do?
Yeah, absolutely. Like literally in early April, we were telling regulators and, well, decision makers on COVID that history shows that in all pandemics you have multiple waves. So even though the caseloads start to go down in New York and Massachusetts, we get them to prepare for the second waves, and we were ahead of others just because we kind of look at the history and try to start modeling of that. Yeah.
Sorry, just trying to mute myself. And linking back to the question about alternative models and methods, I wonder whether either of you could speak a little bit more about this type of kind of uncertainty methods that you might incorporate within the models that we're using. I know quite a few of the listeners from the Risk Institute apply different kind of Monte Carlo analysis or probability balance analysis to try and encapsulate that uncertainty, because obviously when people are screening for new nanomaterials, I guess there are quite a few parameters that they might initially be a bit uncertain about. How do you incorporate that within the models that you're either using, or are there any models that you'd consider that you haven't used yet that might be applicable?
Well, this is actually why I'm really excited about the whole field of resilience. So, you know, in a sense, in the field of risk analysis, we try to figure out uncertainty related to threat and then propagate it through Monte Carlo scenario analysis and kind of output with the idea that you harden the system so it doesn't go down. But in resilience, you really only care about recovery and adaptation. So I don't care how my system gets down in the critical functionality. All I care is about how to design it so it can go up again in the fastest possible way. So from that point of view, uncertainty analysis focus is shifting from the idea that, you know, I don't do Monte Carlo to figure out probability of failure of the system, but rather I'm trying to design it to be recoverable. But, you know, all these traditional methods, you know, Monte Carlo scenario analysis, I know Scott is, you know, a big fan of p-bounds and all that. For me, it really doesn't matter. It's just a question of how much money you have and how much time you have and what decision makers needed. I think that's kind of issue that we have been dealing with for like 30 years. And the challenge is this unknown unknowns, that no matter how much uncertainty analysis you do, something bad is going to happen, then it's going to crush you. How you prepare for that? And solution for that is resilience in my mind.
So I'm not an expert in the kinds of things like the Monte Carlo analysis or anything like that. That's definitely Igor's expertise. For me, the uncertainty has been more at the analytical level. So if I've got a method for measuring, I don't know, for example, how many cells are dead, how accurate is that technique? What's the level of variation from one time to another when I do that, from one person to another? But also, can my measurement technique cover the full dynamic range of the biological effect? So maybe it's only accurate within, you know, from 10% cell death up to 70% cell death, and it's not really telling me much below 10%, and it's not telling me much above 70. Or it could be the other way round, that my measurement technique is actually far more sensitive than what's biologically relevant. And so it's trying to, with the sort of methods that we've been doing the grouping with, making sure that the data analysis is both within a relevant measurement range and a relevant biological range. So mapping out what those two relevant ranges are next to each other to make sure you're doing your analysis within those, because it is tempting when you see a data set to just take the full data set and analyze it, but actually quite a significant proportion of your data set might not be relevant because either it's not biologically relevant or it's not analytically relevant.
That's great, thanks. So before we wrap up, I'm just wondering, does anyone else have any more questions from the audience before we close? If
So
not,
maybe
then,
question
sorry, go ahead.
to Vicky. Vicky, so how do you see, I guess Gracious is very close, so how do you see next steps of that? I guess given kind of focus on safety by design in nano, how do you see all this computational machinery that you develop apply to safety by design?
So there are 3 new projects that have just been funded this year. One called Sunshine, which is coordinated by Ca' Foscari University in Venice. One called Harmless, which is coordinated by one of the Helmholtz Institutes in Germany. And a third one called Diagonal, which is coordinated by a Spanish institution. And all 3 of those projects are to support processes for helping with safe by design of not just nanomaterials. It goes beyond that. It goes to what's called multi-component nanomaterials, so the kind of next generation of more complicated materials. And all 3 of those projects will continue to work on the Gracious project, and they've agreed to work together to do that. So the next step then for that Gracious framework is to make it more widely applicable to a wider range of advanced materials. And that includes the software development as well.
Okay, that sounds great. All right, so I'm gonna wrap up. So thank you very much to both Professor Vicki Stone and Dr. Igor Linkov for their really excellent talks and just the profound impact that they've had on the nanomaterials kind of risk assessment space. And yeah, I guess we're going to be having other talks in other areas as well. So we're expecting a talk on quantum computing. We still have to confirm the date, so we'll be announcing that on the mailing list. There'll be another one on sex robots and sex tech, as well as psychedelic medicine and autonomous vehicles, renewable energies, and the future of food. So please stay tuned, and we'll be back to you soon. So thank you very much again, and enjoy the rest of your day.
Thank you for the invitation.
Thank you.
Thanks,
Thank
Professor Vicki
you.
Stone. Thank you, Igor. Bye.
See you everyone. Thank you.